So, good afternoon to all. I hope all of you had your lunch, and after the lunch, all of you are here for the class. Uh, in the last class, we were discussing about the ultrastructure of the chromosome or the structure of the chromosome under the nucleus. So, in human beings, there are 23 pairs of chromosomes in the human nucleus. And all these 23 pairs, that is 22 autosomes or body chromosomes and 2 sex chromosomes, both of them are in the nucleus. All of them are in the nucleus. Altogether, they are 23 pairs. So, suppose if you open and extend the DNA molecule in each chromosome, if you unwind it, because the chromatin fibers, they have wool and they have become chromosomes. If you unwind them and stretch it in the form of chromatin, it will be about 5 centimeters long. If you link all the DNA molecules in the nucleus, all the 23 pairs of chromosome unwind, then they will become 1.7 to 2 meters long. But the diameter of the nucleus is shorter than 10 millimicrons. Remember, if you take the the entire nucleus is diameter is 10 milligram. Inside this, there is nucleoplasm, there is nucleolus, and these chromosomes also, which are about 1.7 to 2 meters long. So, here by this what we can understand is, the chromosomes are the hereditary material. They contain all the genomic information. That means this genomic information is packed so tightly in the small space of the nucleus inside the cell. Cell is microscopic. Within that, nucleus is also microscopic, and in that you can see all these things 1,000 times smaller than one dot. If you put on a white paper from your pen tip, if you put a one dot, the entire nucleus will be 1,000 times smaller than a dot. Then you imagine the intricacy of this in such a small space. Such a vast information has been being stored there, and the primary structure formed by the powerful compaction. How we are able to pack all this genomic information? What is the primary structure of the chromosome? Where actually these informations are being packed? If you talk about that, probably we can say that this powerful compaction of all the genomic information, you call it a nucleosome. That means this is where our genetic information has been stored in a very compact form. Now coming to the nucleosomes. If the chromatin fiber, you take the chromatin fiber from any nucleus, then what you can do is you can treat this chromatin with some enzyme known as nuclease enzyme. This nuclease enzyme, if you treat this chromatin with nuclease enzyme, that will be broken up into DNA fragments. DNA fragments of the size of around 200 base pairs. 200 base pairs. Base pairs, adenine, guanine, thymine, cytosine, adenine, guanine, thymine, cytosine. Like that, there will be 200 such base pairs they will cut. The chromatin fiber will be broken, or it is made fragments of around 200 base pairs. Now, if you treat this null DNA, there is a small short fragment which is having around 200 base pair with the same nuclease enzyme again, what you will get? you will again get randomly degenerated fragments of DNA. DNA, again this small piece which is of the 200 base pairs has been treated with again the enzyme nuclease. Then you will get randomly degenerated fragments of the DNA. Small pieces. Based on this experiment, one cytologist by R. Korenberg, he figured out the model of a nucleosome. Nucleosome, as I have told you, the powerful compaction of our genome. Nucleosome is nothing but the most powerful compaction of our genome. That is, based on this, he tried to understand, he tried to explain the structure of a nucleosome. When we talk about the ultrastructure of chromosome, we land up at nucleosome, remember. It is a very basic one. Now, if you see that nucleosome is a beaded structure. It's a beaded structure composed of two things. One is the core particle and linker DNA. Core particles and linker DNA, each nucleosome. So here, core particle and linker DNA. And if you go to the details of this, each nucleosome includes around 200 base pairs of DNA. That means first when you treat the chromatin, it was broken into 200 base pairs DNA. So this each nucleosome includes about 200 base pairs of DNA. And what this contains? One, as I have told you, there's a one bead. That's a histone core. This bead is nothing but the histone core. And then an H1 protein. If you see the histone core, that is made out of, composed of 8 molecules of 4 types of histone molecule. One is H2A, H2B, H3 and H4. The by two molecules from each. That means the core is made out of histone core, and this histone core is consists of 8 molecules. That is 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 more molecules of H4 histone proteins. Now, to this histone protein, the DNA molecule binds the core particle. Actually, this DNA, that is the base pairs, DNA molecule binds the core particle in a left-handed helix, not right-handed, but it is anti-clockwise, left-handed helix. And it forms 80 base pairs. It requires 80 base pairs for each circle. What does it mean? There's a histone core. Say this is the histone core. And this is the length of a DNA, which is of 200 base pairs. So this DNA winds on this histone core in a left-handed helix, and to take one turn, the DNA requires 80 base pairs. Each circle, complete one circle, takes what you call 80 base pairs are required. And that means there are about 1.75 circle in each structure. In each structure, what happens? There will be 1.75, one complete circle, and another will be 3/4 somewhere here. And adjacent to the particle linked here, there will be linked by 60 base pair linker DNA. Altogether it is 200. One circle will take 80 base pairs. Second circle is only 75%. That means 75% means that is 80 plus 60. How much? 140 up to here. Then another 60 pair will be like this linker DNA. Probably the next picture will give you the more information. One minute, I'll go for the slideshow. See here, have you noticed this? This is the octameric histone core. Each one where my cursor is moving is the histone core. It is made up of 8 molecules of H1A, H2B, H3 and H4 protein. See, this is the histone core. This is the histone core where you can see. This is the histone core. This thread-like thing, what you call, this is nothing but the DNA fragment. This DNA is of 200 base pairs. They wind in an anti-clockwise direction. See here, from here, they have taken a round like this. One full round means up to here, it will take 80 base pair. Second one will be only 75%. It will up to here. That means 140, 80 plus 60% of 80, 60. That means here it will be 160. Then this length, that is the linker DNA. This is another 60 base pairs. ಏನು ಅರ್ಥ ಆಯ್ತಾ ಇಲ್ವಾ? ಜಸ್ಟ್ ಟೆಲ್ ಮಿ. ಹಲೋ, ಏನು ಗೊತ್ತಾಯ್ತಾ ಸ್ನೇಹ? ಗೊತ್ತಾಯ್ತಾ? ಮತ್ತೊಂದ್ಸರಿ ರಿಪೀಟ್ ಮಾಡಬೇಕಾ? ಬಿಕಾಸ್ ಇಟ್ ಇಸ್ ದ ultra structure of the chromosomes. I am not going to the details again when I take up the chromosome. Here only I would like to finish it off. ಅರ್ಥ ಆಯ್ತಾ ನಿಮಗೆ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಅಂದ್ರೆ ಏನು ಅಂತೇಳಿ? ದಿಸ್ ಈಸ್ ಒನ್ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್. ಇಲ್ಲಿ ನೋಡಿ, ಅಲ್ಲಿ ಫೋಟೋದಲ್ಲಿ ಕಾಣ್ತಾ ಇದೆ. ಒನ್ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಅಂತಂದ್ರೆ, ಸಿ ದಿಸ್ ಈಸ್ ಒನ್ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ವಿತ್ ಹಿಸ್ಟೋನ್ core protein, that is H1A, H2A, H3 and H4. 2 molecules each. They form this yellow structure in kanta ideyala, bead like, that is the histone core. Surrounding that there will be DNA of 20 base pairs which own left helical. ಹೇಳಿ ಇನ್ನೊಮ್ಮೆ, ಗೊತ್ತಾಯ್ತಾ ಇಲ್ವಾ ಪಲ್ಲವಿ? ಸ್ನೇಹ?
ಹಾ ಸರ್ ಗೊತ್ತಾಯ್ತು.
ಸ್ನೇಹ?
ಹಾ ಸರ್ ಗೊತ್ತಾಯ್ತು.
So I can move forward, alwa? Okay, now.
Yes sir.
The DNA compacted, such compacted, is shortened by 7 folds. This type of DNA, then it has been folded 7 folds to form a DNA filament, and that is of 11 nanometer in diameter. It is beyond imagination, alwa? Because 7 folds, after 7 folds, that forms a DNA filament which is only 11 nanometer in diameter, and when it is transformed to a beaded nucleosome chain, it looks like a chain. Can you make out here? It looks like a chain. Then it folds. See, now the chromatin DNA exists in another style. By that beaded nucleosome chain, it is condensed by 6 folds also. You can see both 7-fold DNA and 6-fold DNA when you observe that under the electron microscope. And there is one nucleosome. Actually, we can see the chromatin DNA filament in 30 nm diameter, but this is only 11 nm diameter, overlapped helical structure, and of a beaded nucleosome. So nucleosome appears like this. It is known as pearl of strings also. Alwa, if you make a chain using the beads, DNA filament 30 nm and 11 nm diameter. First one is 30 nm DNA filament, and it is in the 11 nm nanometer. That is the nucleosome. It's not enough here, because even if it is done, you cannot store all the information. Then there will be advanced packaging of the chromosome. even most of the information details unknown so far. So we say that probably it is a serial overlap or pleated like. That means it must be a DNA. It has been in 11 nm filament beaded nucleosome chain. It folds into 30 nm filament, pleat or loop chain, then binds to the site on the nuclear skeleton where adenine and thymine attaches, enriched, and then assembly of chromosome takes place. This probably give you the information. So this is the short, the first you see, this is the short region of the DNA double helix. The width is only 2 nm. Then beads on string, this is what you call the nucleosomes. Beads on string form of chromatin which is only 11 nm. Then it goes for folding, 30 nm coiling, supercoiling, and that is known as solenoid coiling. Actually this type of repeated folding and coiling, it is known as solenoid coiling, and it is, the diameter is 30 nm. We started with a single DNA, see this is just DNA fragments without histone core which is 2 nm, and when DNA means histone and nucleic acid, then we have a histone core, and when the DNA is wound on this histone core, it is about 11 nm. When it further folds, coils, then it becomes 30 nm, so because of solenoid coiling. And this is not enough, even that again further coil, further folds, you can see 300 nm, then there will be condensation, further condensation which becomes 700 nm. Then during the cell division, it is chromosomes or what you call chromatin fibers are folded, then it becomes the chromosome in the last one which is about 1400 nm. It started with 2 nm and ends in the formation of a chromosome which is 1400 nanometer. This is how chromosomes are assembled. If you go back to the ultrastructure, you have to go back to the nucleosome, the beaded appearance. Yes, have you understood this also? Coiling pattern?
Yes, sir.
Okay. Now, in the last class I have told you a little bit about heterochromatin and euchromatin. Especially when the cell is in a non-dividing state or what is known as interphase, that is the G1 and G2 phase of the cell cycle. The chromatin in the nucleus can be sorted as heterochromatin and euchromatin. When the chromatin fiber, actually in the non-dividing cell there is no chromosome, it is only the chromatin fiber. This chromatin fiber in the interphase can show two different reasons. One is heterochromatin and another one is euchromatin. Now, what is euchromatin? I have told euchromatin is the light stained region, and where the transcription is very active. That is the where genes are very active, and it looks like a loose loop, and it takes bright staining under the electron microscope. Even you can cleave this euchromatin by nuclease enzyme at some hypersensitive sites. It's thin, bright staining, looks like loose loop, contains active genes. Whereas the other one, heterochromatin, it is condensed without any, where genes are silent, genes are not active, genes are inactive, and it takes very dark stain. And in other words, we can also say that euchromatin is the active region of the chromosome, whereas heterochromatin is the inactive region of the chromatin. Euchromatin is the active region on the chromatin, and heterochromatin is the inactive region. So it is not wrong if you say the heterochromatin is the genetic lazy region. It is the genetic lazy region and replicated lately, but whereas condensed early. This is called as heteropyknosis. That is, the chromatin being the genetic lazy region, and it is replicated lately and condensed early. Replication was late, but whereas the condensation was earlier, that is known as heteropyknosis. See here, we have, this is the chromatin. You can say the light stained region, they are the euchromatins. They are the euchromatins. And this is the dark stained heterochromatin, that is genetic lazy region, whereas it is the genes are very, very active here. Heterochromatin is dark staining, whereas the euchromatin is bright stain. Now, in heterochromatin, again there are two types, constitutive heterochromatin and another one is facultative heterochromatin. So this constitutive heterochromatin is a heteropyknosed chromatin in each of the cell, and it is located in the centromere. I hope centromere is the region where two sister chromatids attach. Where two chromatids attach. So that is the constitutive. Whereas the other one, facultative heterochromatin, this appears in some of the very special cell types or only during the developing stage. The X chromosomes of female mammalian is facultative. The X chromosome of female human mammals is heterochromatin. Double X chromosome and one heterochromatin called bar body. When the human embryo is developed after 16 days, one X chromosome will be transformed as bar body with dark staining. So that is the facultative heterochromatin. We need not go to that much details. Now here, after studying the infrastructure of the chromosome, let's briefly talk about the structure of the chromosome, because I will not discuss that in detail here, because tomorrow I have to take up the chromosome, study of chromosome separately. Especially in the M phase, that is the mitotic phase of the cell cycle, chromatin will be transformed as chromosome by powerful condensation. By powerful condensation, this long thread-like chromatin fiber will be converted into rod-shaped structure by powerful condensation. They are almost stick-shaped with different lengths. Probably in the cell cycle, in the mitosis, there is the cell division, the metaphase chromosome is the best state to observe. Metaphase chromosome is the best state to observe, and about that I will discuss tomorrow, not now. All those things, primary, all those things I will take. This is how a chromosome appears, all those things I will take. All the entire chromosome I will not touch upon here. Tomorrow when I take up there is a separate chapter, chromosome, there only I will take it. So all these things I will skip now further. Okay, this is about the chromosomes or chromatin fibers. I am not talking about the chromosomes, but it is the chromatin fibers. So as far your syllabus is concerned, during the cell organelle, nucleus and nucleolus. In the nucleus only I have discussed the ultrastructure of the chromosome also up to nucleosome. Now, in the cell organelle, only the thing that is left is nucleolus. Nucleolus. Nucleolus is a spherical suspension in the nucleoplasm. It's a distinct spherical suspension in the nucleus. In the nucleoplasm, we call it as nucleolus. Now, how many nucleolus you can expect? Number of nucleolus is proportionate to the number of genomes present in the chromatin. How many genomes are present in the chromatin? That many number of nucleolus will be there. But generally it is one. Nucleolus, if you analyze it, though it's a suspension within the nucleoplasm, is very rich in RNA. That's the ribonucleic acid, RNA and histone protein. Nucleolus is always rich and it is acidic because of ribosomal RNA, ribonucleic acid, and there are histone proteins. The nucleolus becomes relevant and important for us because it is the site of ribosome synthesis. The ribosomes which you have seen on the surface of the endoplasmic reticulum basically originates in the nucleolus. It is the site. It is produced in the nucleolus, then has to pass through the nuclear membrane and then reach the cytoplasm and get studied on the surface of the endoplasmic reticulum and make it rough endoplasmic reticulum. It is the site of formation of ribosome. This nucleolus becomes a secondary constriction. Nucleolus becomes a secondary constriction on specific chromosome during the cell division. On a chromosome, there will be constrictions. One permanent constriction is there, that is what you call primary constriction, and that is known as centromere. But in addition to that, not on all chromosomes, especially on one chromosome, this nucleolus becomes the secondary constriction and makes that chromosome as satellite chromosome. That I will explain tomorrow when I take up that. The nucleolus is also visible only during the interphase. Nucleolus is also visible only during the interphase, because once the cell division starts, there is a disappearance of the nuclear membrane. With the beginning of the cell cycle, there will be disintegration of nuclear membrane or nuclear envelope. When the nuclear envelope or membrane disintegrates, there will be merging of cytoplasm with the nucleoplasm. There's a merging of cytoplasm with the nucleoplasm. So that's why the nucleolus is also not visible. It becomes secondary constriction in the cell division. So that's why if you want to see the nucleolus, it is only in the interphase cell. The nucleolus, they are spherical area. Generally there will be one or two for each cell, usually. Mostly one, because cells have only one genome, sometimes two. The number and size of nucleolus depending on the cell type and function. What is the size of the nucleolus and what is the number of nucleolus? It depends upon the cell type and function. Suppose if a cell is involved in more protein synthesis and faster proliferation, either more protein synthesis or faster proliferation, more and bigger nucleoli in the cell. The nucleoli will be bigger and more. The nucleolus disappears before the cell division, remember, and again reappears at the end of the cell division. They again reappear when the cell division completes. The major functions of nucleolus is one, as I have told you, ribosome assembly or synthesis of ribosome, and the next one is rRNA transcription. Transcription of rRNA. See, actually the nucleolus appears like this, spherical structure. No membranes cover this nucleolus. Even within the nucleolus, there are three spatial areas which can be seen or understood only with the help of electron microscope. What are these three centers? One is the fibrillar center, FC, that are surrounded by dense fibers. Because of the dense fibers, they have low electric density. These fibrillar centers contain RNA polymerase enzyme. They contain RNA polymerase enzyme and rDNA, which is a naked molecule, rDNA or ribosomal DNA. The second one is known as DFC, dense fibrillar components. Fibrillar centers or components, fibrillar components, and DFC, dense fibrillar component. That's a loop or half loop. It's a loop or half loop which surround these fibrillar centers. Which surround the fibrillar center. Here in this dense fibrillar component, transcription is carried out in the border region of the, it carries out the transcription at the border region of fibrillar center and at the the border region of dense fibrillar component. Third one is granular component. This granular component, in short it is called as GC, composed of 15 to 20 nm particles. Consists of 15 to 20 particles that are RNPs in different manufacturing steps. What do you mean by RNP? RNA combined with protein. RNP means RNA combined with proteins. These are the small particles of 15 to 20 nm, and they take part in different and they are in different manufactured steps. Even nucleolus chromatin can be sorted as heterochromatin and euchromatin. This nucleolus heterochromatin, that is the inactive gene, is also located around the nucleolus. So we call them as nucleolus peripheral chromatin. The heterochromatin, which is always located around the nucleolus, is known as nucleolus peripheral chromatin. Whereas the nucleolus euchromatin is located in the nucleolus, and nucleolus organizing region. It's the center. And that region is known as where rDNA is also located. So this is the ultrastructure of the nucleolus. It has three. One is fibrillar center, dense fibrillar component, and granular component. Here the transcription of rDNA occurs in the fibrillar center. Whereas the granular component contains protein nucleophosmin. It contains protein nucleophosmin. See, this is the granular portion which you can see here in the nucleolus. RNA fibrils. Then, if you look into the chemistry, chemical nature, the main composition of the nucleolus are also protein, RNA, and trace of DNA, that is the rDNA. Protein and RNA and a trace of DNA. This structure is made up of protein and ribonucleic acids. It is made up of proteins and ribonucleic acid. The main function of this is to rewrite the ribosomal RNA, rRNA, and combine that with the proteins. The main function here. Rewrite the ribosomal RNA depending upon the messenger RNA, the gene genetic information, and combine it with specific protein. The nucleolus is generally divided into three parts: fibrous, granular, and matrix, which you can clearly see. Fibrous, granular, and matrix. Here is the granular component, this is the fibrillar component, and this is the matrix. So the nucleolus, it is the region in the nucleus where the portion of the chromosomes which contains gene coding for ribosomal RNA, and they are transcribed into ribosomal subunits. Again, the details we need not go to that. So this is the what I wanted to convey to you with respect to the cell and its function, including all the cell organelles. With this, we have completed. It's a 6 hours. This is 6 hours portion. We have completed all these things. I have taken, I think, 7 hours. I don't know, remember. And then another 5 hours will be chromosomes. And then nucleic acid another 5 hours. Mitosis and cell cycle 5 hours. So we will take all these things one by one in the next class. So in this hello ಕೇಳ್ತಾ ಇದೆಯಾ? Hello, can you hear me?
Yes, sir.
So any doubt? ಇದರದ್ದು ಉಳಿದಿದ್ದು ಅಪ್ ಟು ಎಕ್ಸೆಪ್ಟ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಬಾಕಿ ರೈಟಪ್ ಎಲ್ಲ ನಾನು ನಿಮ್ಗೆ ಕೊಟ್ಟಿದ್ದೇನೆ ಅಲ್ವಾ?
ಹೌದು ಸರ್.
Okay, nucleus within a couple of days I will be able